Dear learners, welcome you all in the course of Biodiversity BBYCT 131. It is a core course. It is for the BSc General. And today I want to discuss very important topic, the bacteria, general account and economic importance. Dear learner, my name is Dr.Siya Ram. I am an associate professor, Discipline of Life Science, School of Sciences, IGNOU. Dear learner, uh, uh, what is the expected outcome of this session? The expected outcome of this session, uh, it is the discuss the history of the bacteria. After completion of this session, you will be able to list the characteristic feature of the bacteria, classified bacteria based on the shape and size, and identify the structure and morphology of the bacteria. After completion of this session, you will be able to describe the various organelles found in the bacteria and appreciate the economic importance of bacteria. Number of the bacteria, they are very useful, and some bacteria, they are very harmful. So we will discuss in detail about the bacteria. So it is the expected learning outcome of this session. My my session is divided into two parts, part first and part second. In part first, we will discuss in detail related to the history of the bacteria or history of the microbiology. And in case of the second part, we will discuss the general account of bacteria, like their morphology, their structure, and other things we will discuss in detail, the economic importance of the bacteria. So I want to discuss one by one. Part first, scientists have found the trace of the bacteria living more than 3.5 year ago, nearly 3 times older than any other known organism on earth. So you can understand that the bacteria is so old, 3.5 million year ago, nearly 3 times older than any other known organism on the earth. Approximately 5,000 bacterial species are associated with the plant, animal, soil, water, ice cap, and harsh environment. Harsh environment means it is a type of environment where the salt concentration and pH salt concentration, it would be very high, and pH concentration will be very high, and so you can say it is the harsh environment. The bacteria, they are able to grow in the harsh environment. And bacteria are omnipresent. You can say the bacteria, they are present everywhere. I have already discussed it. It will present in the on the plant, animal, soil, water, ice cap, everywhere the bacteria is present. They have various morphology and physiology. So they are able to ferment it, and they are able to produce the secondary metabolite, and drugs are there. Their morphology, it would be very different. So we can say it is a omnipresent, have various morphology and physiology. Bacteria, they are able to fix atmospheric nitrogen and degrade organic compounds in the various aquatic habitat, and allowing life to survive. It is a important event of the microbiology. So a lot of important event of microbiology are there. So it is a investigator, number of the investigator are there, events are there. You can say the milestone are there. Year, in 1676, Antony von Leeuwenhoek, the first observed a very little that is known as the animal clues, now they recognized as a protozoa. So a lot of work has done by the Antony von Leeuwenhoek. Edward Jenner, the vaccination with the cowpox to prevent smallpox, means cowpox they are able to prevent the smallpox. So it is a remarkable discovery in field of the vaccination. The cowpox they are able to prevent the smallpox. Another scientist, Louis Pasteur, and they give the idea how to fermentation take place by the yeast. The number of the products are there like wine and beer. A lot of alcoholic products are there. The first time they are able to say the yeast, they are able to ferment the sugar, or ultimately we will get the alcohol. And Joseph Lister, the antiseptic principle in the surgery, means number of the compounds are there, chemicals are there we can utilize when the surgery perform. So like carbolic acids and other antiseptic chemicals are there. He first discovered in 1867. Robert Koch, he first isolated Bacillus anthracis, is a causative agent of the anthrax. It is a very dangerous disease. Ultimately, a number of the it is used, this agent is used as a bioterrorism. Robert Koch, discovery of the mycobacterium. Right now it is an obligation in our society. The first time in 1882 the Robert Koch they have discovered the mycobacterium. Mycobacterium tuberculosis is a very dangerous disease. The number of the person if um, uh, they get disease, or they will die easily. And another scientist, Hans Christian Gram, he discovered the gram staining, or ultimately we can distinguish between gram positive and gram negative bacteria. Sergei Vinogradski, nitrification and chemolithography. And Emil van Ayer in 19 Emil van Waring in 1890, Emil van Waring, the antitoxin for diphtheria. Diphtheria is another very dangerous disease. He discovered the antitoxin for the particular disease that is known as the diphtheria. And Dmitry Ivanovsky, they discovered the virus, the causative agent of the tobacco mosaic disease. And in 1928, in Frederick Griffith, he discovered the transformation. Basically, the DNA they get transferred from one bacteria to other bacteria. So you can say the golden age of the microbiology was between the 1857 to 1910. It is another slide related to the milestone of the microbiology. In 1929, Alexander Fleming it discovered the penicillin is effective against the bacterial infections. Wendell Stanley, he first time crystallized the tobacco mosaic viruses. Selman, Waskman, he discovered the streptomycin. It is another very important antibiotics, or ultimately they will kill the microorganisms. A number of the other scientists are there. They are able to give the various discovery related to the microorganism, like R.J. Seno in the culture 40 million year old bacteria. You can say this bacteria is very old, but we can cultivate the microorganism. So we can grow the microorganism with the help of the different type of the media are there. James B. Capper et al. And circular chromosome in the Vibrio cholerae. And another scientist that is known as the J. Craig Venter, he first large scale environmental genome. He utilized the shotgun sequencing of the Sargasso Sea. These are the various milestones related to the microbiology. So a lot of scientists they are involved to discovery of the microorganism. They discover different type of the drugs are there, and different type of to the food product are there, fermented products are there. So a lot of things are there related to the microbiology. So it is related to the discovery of the microbiology. uh, I want to discuss some very important things related to the history of the microbiology. In uh, Lucretius in 98 to 55 BC, the physician Gerolamo Francescolo in 1478 to 1553 suggested invisible living creature cause the disease. Means there are the some invisible living creature they are able to cause the disease. And Robert Hooke first compound microscope built a first compound microscope and use it to observe the minute compartment in cork slice known as the cell. So these are the various discovery are the related to the microbiology are there. So you can say it is a history, it is a earlier history of the microbiology. In around 1664 he also observed elongated stalk where were fed to the fruiting structure of the fungi. So with the help of the microscope they are able to discover different type of the microorganism like fungi. The development of more advanced microscope and various techniques such as staining and growing of the microorganism made it easier to study of the microorganisms. And I want to discuss related to the pre-20th centuries. Despite prehistoric microbiology did not exist as a science until 19th century. So there is 19th before 19th century the microbiology word is not exist. And in that time beer and wine production were very common, and food were preserved by drying and addition of the salt. So if you want to in that time the food were preserved by to the drying by drying is with the help of the sun and by adding of the salt. The various salt we can use like sodium chloride we can utilize it, or ultimately we can preserve our food. And disease used to spread the reach epidemic proportions, but no one knew their causes. In pre 20th century, some scientists have proven the role of microbes. There are the four scientists are there. They proven the role of microbes in the particular cases, like the Antony von Leeuwenhoek, Louis Pasteur, Robert Koch, and Martinus Beijerinck. So these are the four scientists. They made enormous uh, uh, enormous enormous role. They give the role of the microorganisms which are present, which are available on the earth. So I want to discuss one by one. There are the four scientists, not four scientists, other scientists are there. So I want to discuss one by one. Antony von Leeuwenhoek in 1632 to 1723. Antony von Leeuwenhoek, popularly known as father of the microbiology. He born on 20th 10 1632 in Delft, Netherlands. He ground glasses. Basically, the glasses are there. He ground it, or he prepared the lenses which they utilize for making of the microscope. It can magnify around 50 to 300 time of any object. So these are the very particular discovery are there, very specific discovery are there, you can say, in the field of the microbiology. He was the first scientist to observe the microbe including bacteria, fungi, even he discovered the protozoa also. And he discovered also the capillaries, the blood capillaries which are available inside to the body. He eventually made more than 250 microbes, and he examined the various sample they can collect from the river water, saliva, and feces. He referred to the microscope form life and animal clues and believing them to be a teeny living animals. Another scientist, they have they given enormous discoveries in field of the microbiology, that is known as the Louis Pasteur. First time he disapproved the spontaneous generation theory. In this field, one scientist they give some clue related to the spontaneous to disapprove to the spontaneous generation. It is the generation. It would not be the spontaneous generation. So he gives some clue related to the spontaneous generation. He took meat in the 3 container. So you can see it. These are the one container, second container, and third container. So it is a they took the meat in the 3 container, and one was the uncovered, it is uncovered, and second was the sealed, and third was covered with to the fine gauzes. And here is apply the maggot. The maggot flies and come from the egg of the flies. Another, the maggot did not arise from the second and third jar. He, as egg did not come in the contact with the meat of all. So he give the clear indication of the clear indication against the spontaneous generations. So this experiment is very famous. And after that, the Pasteur disapproved. Pasteur disapproved the theory of spontaneous generations. So in this generation by the experiment. So you can see this experiment by utilizing a figure, and look at this figure. The flask long neck is bent into S-shaped curve and contain are sterilized. So you can see it. Here is the flask are there. It is media. Here is the long neck of flask are there. Is bent into S-shaped and curved, and content are sterilized. And no microbial growth occur if flask remain upright. So you can see it, here is no growth of the microorganism will be there. On bending of the flask, microorganism trap in the neck and come to the contact with the nutrient liquid and grow rapidly. So it is a this experiment is clear indication. It give the clear indication that is the disapprove the theory of the spontaneous generation. It is given by the Pasteur. The bacteria are the cause of the disease reported before Pasteur, but not approved that the germ theory of the disease. So it is with the help of this experiment, the Pasteur disapproved the germ theory of the disease. The Pasteur discovered the microbes ferment fruit and grain to product beer and wine. There are certain microorganisms are there. They are able to ferment or ultimately they will produce the beer and wine, means alcohol. It is ferment or it is produced by the certain microorganism. He also developed the pasteurization technique. It is a very famous technique. So in this technique we can save our milk for longer durations, and we can remove the unwanted micro from juice. So ultimately we can keep it for 2-3 days of your milk without kept in the fridge. So these are the temperature. He utilized 62.8 degrees centigrade for 30 minutes and 71.7 degree for 15 minutes. The pasteurization is a very important discovery of the of a science of Pasteur. And rabies is discovered, it is another very harmful disease are there, that is known as the rabies. Rabies is a disease transmitted to the peoples by bite of the dog, cat and other animals. Even number of the animals like dog, like monkey, like cat and like squirrel, they can transmit the rabies to the humans. The Pasteur developed vaccine by removing the brain and spinal cord from the infected rabbit and dried, pulverized and mixed with glycerin. So basically, Pasteur developed the vaccine of the rabies by utilizing of the certain tissues, and part of the tissues are there. They like spinal cords and brain are there. They utilize it. They dried it, or ultimately this particular tissues they can utilize for the vaccinations, or ultimately they will utilize the glycerin will mix it, or ultimately mixture of these viruses they injected inside to the human, or they are able to cure the rabies. So it is first done by the Pasteur. Another demonstration, Pasteur's demonstration of the immunization. He first time demonstrated how immunization take place by with the help of this experiment. You can understand by his experiment. The experiment number first, it is a old culture of the chicken cholera bacteria. The chicken cholera bacteria is inoculated into the chicken and no sickness are there. Means old, if you inoculated the old culture chicken cholera bacteria, the no sickness will be there. And in case of second experiment, here is the fresh culture, the chicken inoculated earlier chicken, no sickness will be there. And another is the fresh culture, if inoculated into the chicken, ultimately chicken will die. So means this chicken they get immunized with the particular bacteria are there. The first time he gave the immunizations, how immunization take place with the experiment, with this famous experiment. So during 1980, he isolated the bacteria causing the chicken cholera. He found the bacteria could lose their ability to produce the disease, mean virulency will be very less, and attenuated bacteria still stimulate the host and produce antibody to protect against the virulent organisms. Another scientist that is known as the Robert Koch in 1843 to 1910. The Heinrich Hermann Robert Koch was a German physician. He is a great scientist. He is known as the father of bacteriology. Robert Koch was isolating Bacillus anthracis and Vibrio cholerae. I think everyone know that Vibrio cholerae is a very dangerous bacteria are there. Even Bacillus anthracis is also very dangerous bacteria. They can cause the disease like diarrhea. And Koch discovered the Mycobacterium tuberculosis in 1882, and Koch was awarded the Nobel Prize in 1905 for his work on the tuberculosis. And Ferenc Hesse and the wife of Koch assistant Walter Hesse suggested the agar as a solidifying agent in the medium. So it is a history related to the microorganism. How can grow it? If you grow the microorganism, agar is a very important compound. Agar is a solidifying agent we can utilize as a bed. On a particular bed, and the bacteria they are able to grow on the agar. Agar melt at 100 degrees centigrade and solidify at 40 degrees centigrade. So it is a quality of the agar first time discovered by the Ferenc Hesse. And Koch also prepared the tuberculin. It is very important compounds are there. It is a substances that is useful in the diagnosis of the tuberculosis. So tuberculin you can inject it, or ultimately we can diagnose the microorganism. It is Koch's postulate. Robert Koch, they give the postulate a disease suspected causative agent must be found in the every case of the absence. So you can understand through this figure. It is a it is a rat. It is a disease causing rat are there. So in this case, if they get sick, so you can see it. These certain microorganisms are there. So this microorganism you can grow in on a particular media, or you can say grow outside of to the host. When these isolate bacteria are inoculated in healthy susceptible host, host must develop these same diseases, and this same agent must be then re-isolated from the experimental host. So these are the Koch's postulate are there. And another very important scientist which are involved in the field of the microbiology, that is known as the Martinus Beijering. He first isolated the aerobic nitrogen fixing bacteria Azotobacter and Rotonodule, rhizobium capable fixing nitrogen and sulfate reducing bacteria. Both were established the biochemical cycle. Vinogradski discovered the soil bacteria could oxidize iron, sulfur, and ammonia to obtain energy. So these are the number of the scientists who involved in this field. In transition into 20th century, Joseph Lister, he reduced the microorganism from infecting wounds and help an antiseptic that is known as the carbolic acid. So carbolic acid in that time they are utilizing as the antiseptic. Paul Ehrlich proposed the cell blood express in the blood express the variety of the receptor could react with the infectious agent and the initiative them. Alexander Fleming, he discovered the penicillin, and Florey chain and Fleming salesman Waskman is soil microbiology discovered the streptomycin, which is produced by the Streptomyces gracias. And dear learner, I want to summarize my part first. These are the several milestones I have already discussed it. A number of the scientists which are involved in this field, the Antoni van Leeuwenhoek, Louis Pasteur, Robert Koch, Martinus Beijering, and established the role of microorganism prior to the to the 20th century. Dear learner, we will take a short break. Don't go away. We will come back within 2 to 3 minutes.
We wear clothes on a daily basis. But have we ever wondered, where do our clothes come from? Who makes our clothing? And who are the people and the process responsible for the whole manufacturing of our clothing? If you want to know more about this, Indira Gandhi National Open University from the School of Continuing Education has launched a program which is Certificate in Apparel Merchandising. There are many benefits after doing this program. You can become a designer in various fields. It can be fashion designer, apparel designer, costume designer, or even a textile designer. One can also work as a fashion consultant. Research and development is another field which one can take up after doing this certificate course. Going into academics is another perk after doing this program. Getting into fashion houses or fashion studios as an intern is also a very good option post doing the certificate in apparel merchandising. The basic eligibility of the program is